Put simply, cladistics is really just the method of classifying organisms based on common ancestry. It's how we know that we share a fairly recent common ancestor with chimpanzees, and even further back than that, gorillas. We often represent this visually using a diagram known as a cladogram, like the one shown here. You can see that cladograms include something known as a clade, as we have circled in that light grey box there. And a clade is really just a group of organisms that consist of a common ancestor and all of its lineal descendants. So essentially, if you trace all the organisms in a group back to a single common ancestor without leaving out any descendants, then you have a clade. This diagram really helps to illustrate this concept. The first one shows a proper clade. All the organisms share a common ancestor and no descendants are left out. On the other hand, we also have these examples that are labeled not a clade, and these show groups that either leave out some descendants or don't include all the members that share a common ancestor, which breaks the rules of cladistics. From viewing this, you might be able to realize there are a couple of assumptions that we make when we're using cladograms or just cladistics in general. Firstly, all organisms are related by descent from a common ancestor. So there exists one organism, usually called the last common ancestor, from which every living species on Earth descends. You would see the species at the very start of a cladogram if it was showing the entirety of Earth's lineage. Second, the branching pattern follows something known as a bifurcating process, which is probably a term that you haven't heard before and can sound a little confusing, but all it really means is that one species can split into two, and it can't merge back together. And that probably makes sense based on what you know. You probably have never observed a species forming back into one. And that's why the shaded region in the right of the cladogram is not a clade. These organisms don't have a common ancestor included in the shaded area. Thirdly, traits can change over time across different evolutionary branches. And that just basically means that different lineages shown in this diagram don't describe all the same species with the same traits. They're each unique from each other. It's important that you're able to remember these three assumptions. However, you can actually infer them a lot from just looking at this diagram and considering the assumptions that might underlie it. If it's not easy for you, just feel free to focus on actually just memorizing the list. Again, though, it is important that you're able to memorize them. So I'm just going to get a drink. You can take a second to look at those if you'd like. Alrighty, so how do we actually go about making these diagrams? We actually use something called molecular homology, and molecular homology basically refers to the similarities in genetic material of different species that arise from a common ancestor. We can actually do this by comparing the nucleic acids that appear in the genes of different animals, like I was discussing earlier. In this case, we're looking at the number of base sequence differences in a specific gene called the cytochrome C gene across a couple of different species compared to humans. And you would expect that the fewer the differences we observe, the more closely a species is actually related to humans. For instance, we see that the rat has 34 differences, the chicken has 58, and the yeast has 110 in the specific gene, at least when compared to humans. So these molecular differences can actually be used to build a cladogram, which basically visually represents evolutionary relationships. The rat is more closely related to humans than the chicken or yeast, and the chicken is more closely related to humans than yeast. This method of constructing cladograms provides a really quantitative approach to approaching evolutionary relationships by comparing genetic divergence. It's a lot more reliable, again, than things like morphology or methods of reproduction. And the greater the number of genetic differences, the further back in time those species branch off from a common ancestor, which allows us just basically to reflect their evolutionary differences more clearly.